Good afternoon and welcome to this year's Henry Russel Awards Program and Lecture. I'm Mike Solomon, Dean of the Graduate School. This is a very special event on the university's calendar. It's a day we honor a colleague with the highest distinction this institution can bestow, the Russell Lectureship. The 2026 Henry Russell Lecturer is David Ginsburg, whom President Domenico Grasso will introduce to you shortly. I would like to begin by sharing with you the history about the namesake of the Russell Award. Henry Russell earned 3 degrees from the University of Michigan, a Bachelor of Arts degree in 1873, a law degree in 1875, and then a Master's of Arts degree in 1876. Russell was born in Detroit and remained in Michigan after graduation, where he was a successful lawyer and prosperous businessman who was involved with the Michigan Central Railroad, Michigan State Telephone Company, Union Trust Company, People's State Bank, and Detroit Steel Products Company. In February 1920, while in New York City, Russell took ill and died at age 67 from pneumonia, just as he was preparing to travel to Europe to claim the body of his son, Lieutenant William M. Russell, who was killed in aerial combat during World War I. In his will, Henry Russell left the University of Michigan a $10,000 bequest to create an endowment. His one stipulation was that the income from the endowment should be used for additional compensation to members of the instructing staff. In May of 1925, the Regents established the Henry Russell Lecture as a way to recognize a senior member of the faculty with an honorarium of $250 funded by Russell's endowment. In addition, $250 was dedicated for an additional award to honor a faculty member below the rank of professor for conspicuous service to the university. Although the funds from Russell's bequest have long ago run out, the university continues to honor its most accomplished faculty with the award in his name. I am honored to be involved in today's ceremony to recognize our outstanding faculty members. I would now like to invite Provost Macaulay to present the Henry Russell Awards to 4 faculty members. Thank you.
Thank you, Mike, and welcome, everyone. Last night we had the opportunity to win a national championship in basketball. That was really exciting. But today we get to welcome and celebrate an international champion scientist, Dave Ginsburg, and top leading scholars in our fields. And to me, that is just an academic celebration that I so appreciate. And I thank you all for joining us. This event is really always the high point in our academic year, and it's an honor to share the stage with faculty and with all of you in attendance. I am going to start by introducing our first awardee, Professor Solomon Adira, and ask him to come and join me on the stage. Professor Adira directs the Energy Transport Laboratory, where he leads pioneering research at the intersection of heat and mass transfer, fluid structure interactions, and advanced thermal management. His work focuses on phase change-based cooling strategy for power electronics and lithium-ion batteries, using micro and nano engineering, wetting science, and the manipulation of droplets and bubbles to improve heat transfer on engineered surfaces. Through innovative design, fabrication, experimentation, and modeling, his laboratory is helping overcome critical challenges in next-generation electronics cooling. Professor Adira earned his bachelor's degree from Georgia Institute of Technology and his master's and doctoral degrees from MIT, all in mechanical engineering, followed by postdoctoral work at Harvard, before joining the University of Michigan in 2020. A dedicated teacher and mentor, he prepares future thermal engineers through both the classroom and hands-on laboratory research, while fostering outreach and engagement with students at every level. Professor Adira, in recognition of your outstanding contributions to advanced thermal science and engineering education, the University of Michigan proudly presents you with the Henry Russel Award. Okay, next, will Professor Kevin Jeffrey Field please join me on the stage? Kevin Field is nationally recognized for pioneering research in next-generation energy materials that are advancing the safety of nuclear power reactors and accelerating clean, sustainable energy worldwide. At the University of Michigan, he leads groundbreaking work on accident and radiation-tolerant materials for nuclear power generation, including the development of iron, chromium, aluminum alloys. His scholarship includes 2 patents, 82 refereed journal articles, and 31 research grants, including 5 from the U.S. Department of Energy. As chair of the undergraduate engineering program, he has also helped modernize the curriculum and expand key student learning experiences across the College of Engineering. Professor Field earned his bachelor's degree with honors from Michigan Tech and his master's and doctoral degrees from the University of Wisconsin. After 6 years at the Oak Ridge National Laboratory, he joined Michigan and quickly emerged as a leader in both research and engineering education. Professor Field, in recognition of your exceptional contributions to nuclear energy research and to the education of future engineers, the University of Michigan proudly presents you with the Henry Russel Award. Okay, will Professor Teresa Rogers O'Meara please join me at the stage? Teresa O'Meara studies the relationship between fungal pathogens and their human hosts to understand how fungi colonize and cause disease, an increasingly urgent threat to human health. Her research focuses on highly transmissible drug-resistant fungal pathogens and uses big data, functional genomic screens, and innovative genetic and computational approaches to help prevent invasive infections and develop new vaccines and diagnostic tools. Since joining the University of Michigan in 2019, she has received continuous funding from the National Institutes of Health, published 22 peer-reviewed articles, and delivered 22 invited talks. She's also a dedicated teacher and mentor to students across Michigan medicine. Professor O'Meara earned her bachelor's degree with honors from the University of Chicago and her doctorate from Duke, followed by postdoctoral work at the University of Toronto before joining us here at Michigan. Professor O'Meara, in recognition of your fundamental research on drug-resistant fungal infections and your commitment to training future scientists and physicians, the University of Michigan proudly presents you with the Henry Russel Award. Okay, well, Paige Sweet, please join me at the lectern. Paige Sweet studies gender-based violence and the politics of victimization, with a particular focus on how domestic violence has been reframed from a problem of patriarchal harm into a medicalized issue, and the consequences for victims asked to manage that burden. Drawing on archival research and life story interviews, her work illuminates how institutions and expert systems shape intimate relationships and the lived realities of survivors. Her book, The Politics of Surviving: How Women Navigate Domestic Violence and Its Aftermath, received 2 Best Book Awards from the American Sociological Association, and her work on gaslighting was featured by Scientific American as a top story. She is also a deeply respected teacher whose courses are known for their rigor, equity, and support for students. Professor Sweet earned her bachelor's degree summa cum laude from Washington University in St. Louis, followed by her master's and doctoral degrees from the University of Illinois, Chicago, and postdoctoral work at Harvard before joining the University of Michigan in 2020. Professor Sweet, in recognition of your profound scholarship and your commitment to improving the human condition, the University of Michigan proudly presents you with the Henry Russel Award. Let's have another round of applause for these great scholars. And with that, I would now like to welcome our president, Domenico Grasso, to the stage to present our 2026 Henry Russel Lecture. President Grasso.
First, I do want to follow up on what Lori said, because we were both in Indianapolis yesterday, and we did win a national championship, and she beat me to the punch here. But I was trying to figure out how we could get confetti to come down on all our laureates here today, but that wasn't going to work out like yesterday. It was a very special day yesterday, and today is an equally special day on the academic side. And I was very privileged to talk to thousands of alums at our gatherings in Indianapolis, and I underscored more than once how we are outstanding in both athletics and academics, and we're known for academics ahead of athletics. But it was a great experience yesterday in Indianapolis. And I want to thank Provost McCauley for her wonderful emceeing up here of the junior Russell Award winners. This is our way of recognizing and celebrating a faculty member of exceptional achievements in research, scholarship, or creative endeavors, with an outstanding record of distinguished teaching, mentoring, and service to the university and beyond. It is our honor today to present this year's Henry Russel Lectureship to Professor David Ginsburg. David, would you please join me? David Ginsburg is the James V. Neal Distinguished University Professor of Internal Medicine and Genetics, the Warner Lambert Park Davis Professor of Medicine, the Professor of Human Genetics and Pediatrics in the Medical School, Research Professor in the Life Sciences Institute, and much more. It sounds like he should have his own zip code. He is a role model physician scientist who leads a laboratory that investigates the fundamental biology and genetics of blood clotting disorders. Research discoveries made in his lab have advanced science and enhanced lives. Dr. Ginsburg earned his BA at Yale and his MD at Duke, with both postdoctoral training at Harvard Medical School. Last year marked his 40th anniversary as a Michigan faculty member, which has been our good fortune. And interestingly enough, I had the good fortune of flying to Washington with David, and we spent some time, because the flight was significantly delayed, chatting. And I found out that he's been here 40 years, but we've only paid his salary for 38 years because he's been an HHMI investigator the other 38 years, which is truly our good fortune here. He studies the molecular pathogenesis of several bleeding conditions, including the von Willebrand disease, the most common inherited bleeding disorder. His discoveries have led to 2 medications approved by the FDA. His 26 graduate students and 53 postdoctoral fellows are leaders in their field. That's just incredibly remarkable and hard to imagine. He is an elected member of the National Academy of Medicine, American Academy of Arts and Sciences, National Academy of Sciences, and the American Philosophical Society. And I apologize that I lost my voice yesterday screaming in Indianapolis. I think it's most fitting to hear from colleagues who nominated him for today's honor, and I quote, "His stunningly successful scholarly research is accompanied by outstanding teaching in the classroom and the clinic, thoughtful mentorship of scientists and physicians, and an extraordinary level of service to U of M and the scientific community at large. He truly embodies the spirit of the great honor of the Henry Russel Lectureship," end quote. David, it is as one of our nation's foremost scholars in molecular genetics of bleeding and clotting disorders, you have advanced medical science through research discoveries and new clinical therapies, trained generations of scientists, and served this institution with distinction. In recognition of your extraordinary achievements and selfless leadership, the University of Michigan is proud to present you with the Henry Russel Lectureship.
Sure.
Please join me now in welcoming the 2026 Henry Russel Lecturer, David Ginsburg, as he delivers his talk, Genetics of Blood Clotting, from Bedside to Bench and Back Again. David.
Thanks very much. Well, thanks very much for that really gracious and embarrassing introduction, and it's such a wonderful honor to be here to give this lecture. And I guess the part of it I have to accept is this means I'm getting old in some way or whatever. I'm senior now, but anyway, it's an enormous honor to be here. Let me see if I can find the slide advancer. Where did that go? Thank you. All right. So here's the title of my lecture, and a couple of points I want to make today. I mean, the major sort of messages I want to give, 2 really important messages I want to get across. One is this connection as a physician scientist between the patient bedside and the basic research lab. And the other is the impact of mentorship in both directions, both being mentored and then paying it forward and mentoring others. So just a quick summary of what you've already heard about from Domenico, but I started out, my very first lab experience was as an undergraduate at Yale, and I worked in the lab of Joan Stites, who is an absolute giant in science. I've worshipped Joan my whole life. We're good friends, but I was his second undergrad. I've had incredible luck with mentors, blind luck. I was the second undergrad in Joan's lab. That's not what I looked like at the age of 18 in 1972, but I realized we didn't have cameras back. You didn't have cell phones back then. And who was going to bring a camera into the lab? So the only pictures I have are from many years later when we were at a meeting together. I went from there to Duke, as you heard. Did my clinical training in medicine and hematology at the Peter Bent Brigham. And as part of that hematology training, I had to spend a month doing pediatric hematology at Boston Children's, and the attending that month just happened to be this guy, Stu Orkin. And there was no PubMed. I couldn't go look him up, but we got along well. I said, "Can I come work in your lab?" And Stu is a giant in science. Some of you may have heard about curative therapies now for sickle cell anemia. That all comes out of work in Stu's lab. Well, I was Stu's second postdoc. And then I came to the University of Michigan, promised my wife, 4 years or so, and then we moved back east where our families were. Well, That was 41 years ago, and here we are. This is the only job I've ever had in my career. I'm in that faculty picture all the way off to one side. That's in front of the Simpson Institute, which a few of you may recognize, but that's where my first lab was when we first moved here. And it's been an incredible privilege to be here, and you know, it's been wonderful. So I got to start out, I thought, to give you some background, because not everybody is a scientist or a hematologist in this audience. So first the first course we're going to do is Biology 101. And all you need to know here is that all living things are made up of cells, and billions of cells, and in organisms like humans, they all have a nucleus, and they've got DNA in there, and that DNA makes up the chromosomes and carries the information to allow us to make the proteins that do all the work and make us what we are. So, you ready for the exam? That's Biology 101. All right, now we'll go on to Advanced Biology 201. And here, this is genetics. So, in humans, we have our DNA is put in these little units called chromosomes, and we get 23 from each of our parents. We have 23 pairs, 46 total. And it's about 3 billion bits of DNA information. It carries all the information to make the genes and the proteins that make us what we are. In humans, we've got about 20,000 of these different genes. And these genes vary in size from a few thousand of these DNA letters to millions. When I started out, you know, I had training in hematology and had me interested in blood diseases, and this connected to the lab for me. I got interested in blood clotting, and this is now the last class you have to pass, and that's Blood Clotting 401. Blood clotting is a really important process in all organisms that have a circulatory system and blood moving around under high pressures, because if you get injured, the blood will all leak out, and that's not a good thing. And if it clots too quickly, you get into trouble. You have heart attacks and strokes and things from too many blood clots. So this very complex system has evolved to precisely regulate this, and a lot of different genes and proteins that do this. And perhaps more than any other part of medicine and biology, our understanding of this process has come completely initially from the study of human patients deficient in each of these factors, and they'd have a bleeding disease because they were missing one of these key factors, or a blood clotting disease, and that's how these were all defined. And there's sort of a cascade that one activates the next, the next, the next, from a teeny bit to amplify the signal and make the blood clot where you need it, where there's been an injury. And one of those key proteins is a protein called von Willebrand's factor. That's VWF in this slide. And it's this big protein, carries around another important protein called factor VIII. And when there's an injury, it helps bridge these things called blood platelets to the blood vessel wall and make that initial clot that starts things off. And when I started in the lab, von Willebrand's factor was sort of a mystery. We didn't know much about it. These were early days. Cloning genes was a really big deal. That's what got me my first job here, because I learned how to do that. And I cloned the cDNA for von Willebrand's factor, and this was a lot of work, you know, determined the sequence. Nowadays, that takes about 3 nanoseconds at the computer. You just pull the sequence out. You know, it's incredible how this has progressed. But this allowed us to work out what the gene looks like, and then to go look at the patients who have different types of von Willebrand's disease and figure out what changes they have that cause that type, et cetera, et cetera. And when I moved to Michigan, I had this great skill of cloning genes, cDNA cloning, and a colleague here, Tom Gallardo, was working on another blood clotting protein called Pi1. He said, "Hey, David, do you think we could clone that?" And so that was my first paper on my own at the University of Michigan, where we did that, and later on used that sequence information to identify the first set of patients that had a deficiency in this protein, and they had a bleeding disorder. We're still studying that protein, and now it's just amazing. I mean, one of the points I'd like to get across is how science has changed and progressed from the time I started. Back in those days, the idea that you could make a recombinant protein was really pretty incredible. Well, in this experiment here, Laura Haynes, who led this work in my lab, looked at every single possible single amino acid change you could make in that protein. It's something called deep mutational scanning. So along the x-axis is each of the 400 some odd amino acids in this protein. And on the y-axis is every of the 20 possible amino acids. We substituted each one at each of these places, made the 8,000 possible different proteins, and one big experiment figured out which ones made it not work, which ones made it work better, and we've been doing studies like that. And again, when I was starting out, if you would have told me that this would be possible someday, we would have said, that's completely ridiculous. And it is amazing. It's so much fun, the things we can do in the lab now that we couldn't have dreamed of just a few years ago. Well, another one of these things was something called positional cloning. This was a big advance that came out shortly after I'd moved here on the faculty. One of my close friends and colleagues, Francis Collins, led the Genome Project, and actually one of the first things Francis did in the lab here was positionally clone the gene for cystic fibrosis. And basically what this meant was you would start out with some families with a genetic disease, and you would trace how the DNA was being inherited in the family. And if you looked at the people who had the disease, what parts of the DNA had they all gotten in common, and you could narrow down where the gene was and eventually get to it. It was amazing that you could even do this. And when we first started talking about it, we all thought this would be impossible. And now, of course, it's totally routine. The very first few genes were teams of hundreds of people working together to do it. Now, one undergraduate who wanders into my lab by mistake could sequence a couple of genomes in the afternoon. Oh, there it is. It's a totally different thing. And this is sort of summarized here. When I was starting out in the mid-1980s, there was one human disease for which you knew the involved gene, and that's hemoglobin and sickle cell anemia and thalassemia. That was the only disease for which we knew the responsible gene. Today, that's close to 7,000 different diseases and about 5,000 different genes. It's astounding how that has changed. But we've used these methods in my lab to identify several of these genes. ADAMTS13 is a very important protein that works on von Willebrand's factor, and if you don't have it, the von Willebrand's factor is too sticky and you get this terrible blood clotting disease called thrombotic thrombocytopenic purpura. And it turned out, by looking at patients who had an inherited form of that disease, we could trace down the gene for this protein ADAMTS13. And the remaining time, I want to tell you about another disease gene we found that way that then led into a large part of the work my lab's done over the past few years. And that's a disease called combined deficiency of factor V and factor VIII, or F5, F8D on this slide. So what is that? This was a very curious disorder to hematologists. It's an autosomal recessive disorder. It means there's one gene in our genome out of the 20,000 or so that if both copies of it don't work, you have the disease. Well, what is it? And it was so peculiar because the level in the blood of factor VIII, that's what's missing in hemophilics, or factor V, another blood clotting protein causes another bleeding disorder, both of them were down to about 10% of normal. Yet they're made in different types of cells. Their levels in the blood are 100-fold different. How could they both drop to 10% of normal? We had no idea. Well, that was the beauty of positional cloning. We tracked down the gene and found it using these positional cloning methods. Again, wouldn't do that today. Today, you just send off their sequence for $500. You get a whole genome sequence, and there's your answer. But we found a very curious gene. At the time, it was called ERGIC53, which was for Golgi intermediate compartment 53 kilodalton protein. Nobody had any idea what it did, but in the cell, it sat in this place called the ERGIC between the two organelles in the cell, the ER and the Golgi. And now it turned out, we found by this genetic trick that if you didn't have that protein, this is what you got, deficiency of these two blood clotting proteins. So this raised all kinds of curious questions. Mutations in this gene explained about 70% of patients. We then did positional cloning in the other 30% and found another gene. This was a gene nobody had any idea what it does or did, and it was named MCFD2 by the official gene naming groups. That stands for multiple coagulation factor deficiency number 2 gene. And it turns out these two proteins sit together in the ER and form this complex and are associated with each other, which was clearly telling us something. And one of the major points I want to make here, and what I'm going to tell you next, is that part of what drives us as scientists and as physician scientists, we'd all love to cure cancer or cure any other disease if we could, but what drives us often is just the fundamental scientific question, the gee whiz, how does that work? And that sort of, we talk a lot about going from the bench to the bedside, taking a scientific discovery and reducing it to some new treatment. But what's driving a lot of us is this, how does this work? Sure, I'd love to make something clinically useful out of it, but gee whiz, how's this going on? How does this work? And that's what gets us up in the morning and brings us into the lab. So here we discovered this thing that sits in the endoplasmic reticulum in the cell, and I vaguely knew what that was from college biology and all, but I really didn't know much. I had to learn a whole new thing. Well, it turns out that in our cells, we make a lot of proteins that we secrete that go outside the cell and then go do their work somewhere else. In fact, about 30% of the genes in our genome encode proteins that get secreted from the cell. And there's a whole system for how that works. They first get put into this ER or endoplasmic reticulum, and then there are these tubules and vesicles that move into this next thing called the Golgi, and eventually out of the cell. Randy Schekman, who's a collaborator of ours who figured this all out in yeast, and won the Nobel Prize for it, and taught us how to study this in our cells. And what we had discovered by this genetics, never would have guessed this was the case, is that this LMAN1, ERGA53 was renamed LMAN1 by the official gene keepers. That's lactose mannose binding protein number 1. But LMAN1 and MC52 are a complex, and they sit there in the ER. And inside the ER, they recognize the protein that has to get secreted, factor V or factor VIII. And outside, they recognize these things called COP2, this machinery in the cell that helps you move things out. And we had just sort of fallen into this. Well, it turns out in yeast, when Randy worked this all out, there's this protein SAR1 that brings these other two, Sec23 and 4 in, and then there's an outer coat and all that. Well, in humans and in mice and all mammals, we have 2 SAR1s and 4 Sec24s and 2 Sec23s. And that raises the interesting, well, what are these things all doing? Why do we have so many of these? Well, in humans, we know there are few human diseases that are associated with deficiency of these proteins, and they're listed here, only a few. Most of these proteins, we have no idea what happens if you don't have them. Well, right around this time, there was this great new methodology where you could knock out any gene you wanted in a mouse. You could make a mouse deficient in anything you wanted. And I went and looked. There was a catalog of stem cells of mice that had genes knocked out, which you could easily make a mouse from. And all of these genes were in there, and no one was doing anything with them. So I couldn't resist, and we knocked them all out and looked. And it was startling what we got. We got very different results in the mouse than we did in humans. Almost all of these were lethal in the mouse, where some caused diseases in humans. I'll just very quickly tell you about 2 of these, Sec23A and Sec23B. Very similar proteins, come from a single protein in yeast. If you're missing Sec23B, you get a very unique abnormality in your red blood cells in humans, a unique kind of anemia, congenital dyserythropoietic anemia type 2. And that's curious, but in the mouse, their red cells are totally fine, but they totally destroy their pancreas. What's going on? I thought we're just big mice. I mean, how could this be so different? Well, Rami Koryati, who's sitting right back there, was a trainee in the lab now. He's now a very successful established associate professor here. He figured out that there's a difference in which form is expressed in which cell type, so that in humans, Sec23B is the major form in the bone marrow, whereas in mice, Sec23A is the major form in the bone marrow, and Sec23B is the major form in the pancreas. And that could explain all this, and he proved this really by making a unique mouse where we substituted the Sec23A gene for the Sec23B gene. So those mice can only make Sec23A, but they make it all the places a mouse would normally make Sec23A and Sec23B, and that totally fixed the problem in the pancreas. So really proving in the mouse that they're quite equivalent. And the basic idea from evolutionary biology now is that genes in our genomes, as we've evolved and from throughout evolution, are constantly duplicating, making an extra copy. And most of the time, one of those duplicates picks up DNA changes because you don't really need two, and it becomes what's called a pseudogene and eventually disappears. But every once in a while, it will pick up a new function, or it'll divide up the function. So if this is what the original gene did, each one now does part of that job. Now you need both, and then evolution will keep it around. And what we'd found out is that for the Sec23 genes, it was that last thing. They divided their function in terms of which cells they worked in. And this has some interesting implications, potentially therapeutic ones that I'll come back to at the end. And just to go through quickly, we've done a very similar thing with SAR1A and B. SAR1B causes a very unique defect in cholesterol and lipids in people. In mice, it's very early in the embryo. They all die. V. Tang, who was a graduate student at the time in the lab, did the same experiment that we talked about with Sec23. We swapped in the sequence for SAR1A into the location of the SAR1B gene, and it completely rescued the mice. Again, showing that these two genes really do similar things. It's just where they're made that explained everything. So we learned a lot from all this, that there's a very significant and maybe complete overlap in function between the two different versions of Sec23 and the two different versions of SAR1. We've got some similar data for some of the other genes, and that it's a shift in when they're made and in what cell type that explains it. And this raises an interesting idea, because perhaps we could take a patient with one of these diseases, say from not having Sec23B, if we could turn on Sec23A in their red blood cells, that ought to fix the disease. Or turn on SAR1A in the intestinal and liver cells of a patient with Anderson's disease, and that would cure that disease. And so this is, as some of you may know, sickle cell anemia you can now cure by turning on fetal hemoglobin in the red blood cells where sickle hemoglobin is normally made. And this would be analogous to that. Now, finally, let me just tell you a little bit about one other curious one here, and this is Sec24A. This was the one mouse who was totally fine. We couldn't find anything wrong with the mouse. One of those mice in the picture has no Sec24A, the other's normal. I don't know which is which. Totally missing Sec24A. One of the students in the lab who was studying this mouse misread an old paper and tested cholesterols in his mice. I said, what do you do that for? This is nothing. And I explained why. It turned out the mice said their cholesterols were half normal. So, sometimes you get the right answer for a funny reason. Their cholesterols are half normal. Well, how did that work? A brilliant postdoc in the lab, Xiaowei Chen, now has his own very successful and productive lab in Beijing, made the astute observation that these mice had very low levels of, reduced levels of PCSK9. This is a regulator of cholesterol. And in fact, antibodies against it, so you can reduce the levels, is being used, FDA approved, to treat high cholesterols. And for some reason, the level of PCSK9 is about half normal, though the gene is working at the same rate, but the protein suggests that maybe it's not making its way out of the cell. Well, PCSK9 is sitting here inside the endoplasmic reticulum. How could it know that Sec24A out there and not B, C, or D? It implied another cargo receptor. Well, we knew it wasn't that LMA1 MCFT2 that we'd previously discovered, because those patients have normal cholesterols. What could it be? Brian Emmer, who's also now on the faculty here, an assistant professor, did another incredible experiment we couldn't have dreamed of when I was starting out. Using this CRISPR stuff, he did what's called a whole genome CRISPR experiment. In one experiment, he knocked out every single gene in the genome. Each cell had a different gene knocked out in a huge vat of cells. And then he could fish out which ones couldn't secrete PCSK9 anymore. And he found one clear answer, nothing else, a gene called SIRF4. And he had discovered the cargo receptor for PCSK9. And this has turned out to be really a lot of fun and very interesting. If you knock out SIRF4, make a mouse with no SIRF4, it's an embryonic lethal. The mouse dies during embryogenesis. But if you just knock it out in the liver, the mice are fine, but they have shockingly low cholesterols, about 10% of the normal cholesterol, and which again suggests potentially a therapy. Nothing we're going to do, but if some company wants to do it someday, that'd be great. So we've learned a lot, and I don't have time to talk about it, but we're doing a lot of cool new experiments now in cells to try to figure out, well, what are all the genes and proteins that depend on each of these cargo receptors? What's their repertoire of proteins? It looks like LMA1 MC52 has very few, factor V and factor VIII, and a few other things, which is why probably those patients, they bleed, but there's not much else. SIRF4 turns out to have tons of cargos, and we're very interested in trying to figure that out, trying to figure out if there are other cargo receptors that do different things, and a lot of people doing those experiments are sitting right there, and they can tell you some of the details. But again, I want to just end with the point that the real fun and exciting thing is very hard to explain to the general public, but as scientists, what gets us up in the morning and gets us in, and that we're sitting there, and I know some of you can agree with this, is that, gee whiz, how does this work? How could it be that these patients have these two proteins reduced? How could it be that this mouse has a low cholesterol and nothing else? And figuring that out is just so much fun and so exciting. And it's really that bench to bedside is really one of the real joys of this business, and an incredible privilege that we get to have. There may be some opportunities to go back from the bench to the bedside, cholesterol drugs by targeting several of these things, anticoagulants, this idea of increasing one of the paralogs of a gene to make up for the absence of the other, but we'll see where that all leads. And finally, I want to end with just a word about mentoring. I am eternally indebted to my two mentors, Joan Stites and Stu Orkin, who introduced me to science. Joan took me to my first meeting. I wrote my first paper with her. We're still good friends. I worshiped her my whole career. And similarly for Stu. And it's just so much, so great to still be there. Now, and particularly in this phase of my career, I get enormous satisfaction and pleasure out of seeing my trainees, folks here, a few others in the audience who used to be in the lab, and watching all the great things they're doing. And there's this old saying that the real impact you have in science isn't anything you do yourself. It's all the people you train, whose own contributions are going to so far exceed anything I've done. And it's just so rewarding to watch them as they're starting off on this same voyage. And it is really a unique privilege we have being here, and getting to do what we do. I remember one of my colleagues who does what I do telling me years ago, isn't it amazing that they pay us to do this? Or when I talk to some of my friends about, you can ever retire? And I tell them, well, I think what I want to do when I retire is this. And we really are incredibly lucky to be able to do this. And with that, let me just end with a call out to the lab. And this is just, I can't put all the trainees up here, and I feel badly about people I'm leaving out, but on the left are the people whose work contribute to the stuff I talked about today, and there's a lot of things I didn't cover, and the current lab members, a couple of key collaborators, and a picture of folks in the lab right now. And I'll stop there and be happy to take any questions if we have time.
Thank you very much for your lecture, Professor Ginsburg. I'm happy to moderate the question and answer session. We have microphones that will pass, and for those of you that are online, you also have a way to register your questions. So the microphones are here. I'm happy to lead with a question as you're thinking of yours. I was really enthralled by this undergraduate that wanders into your lab and is kind of cloning those genes, work that took you a couple months. And I was wondering, from your vantage now, that's just a remarkable thing to have seen that sea change. If you were to try to look ahead, What is the
Oh,
thing
wow.
that takes 2 months in the lab now that you would have a hope that 20 years from now would that undergrad is going to be able to do?
Well, Mike, that's a fantastic question, and I can tell you, based on my past experience, my batting average at predicting the future is around 0%. I mean, again, if you would have, my very first scientific paper with Joan Stites back in the early 70s, I worked for a year and a half in the lab. We sequenced 4 base pairs. The first 4 base pairs of the ribosomal RNA precursor in E. coli. DNA sequencing hadn't been invented yet. 4 base pairs in a year and a half. By the way, it was ACUG, for those of you who always wondered. And now, again, send off for $500, you can do 6 billion base pairs in an afternoon. I mean, it's totally astounding how this has changed and the things you can do. And I just can't, if you would have told us then, I could never have imagined this would have seemed impossible. So I would predict it's the same thing now, that what's going to be true in another 10 years is, I could never possibly guess now. What I see happening already is a lot of things we used to do ourselves at the laboratory bench. I talk to this with my trainees all the time. We now send out somewhere. I mean, I'm talking with one of my postdocs about an experiment we want to do that, in the old days, like say a year and a half ago, we would have done that ourselves. Now, you pay a company to do it. Actually, the biotech industry has driven a lot of this because, making a mouse with a particular genetic change. In the old days, we'd do that ourselves. That was a year and a half, 2 years of work for a fellow in the lab. Now, you pay a company and they do it for you and you get it. And I think more and more, we'll be sitting in an office, sending the experiments off to a lab to be done for us, getting the data back, and then writing up the paper. And in some ways, it'll be sad not to be at the bench, because it sure is fun to be at the bench. But on the other hand, the things you'll be able to do, and it's going to be a very exciting time. I often tell my trainees I'm a little jealous of them for starting their careers now, because the things they could do, the 3 billion base pairs as opposed to the 4 base pairs in a year and a half, 3 billion in an afternoon. That's quite a scale of change. And it's just an incredibly exciting time. And I wish I knew. Part of me would love to go into a time machine and go forward and see what are they doing 20 years from now, And I just can't imagine I can guess.
Yeah. Wouldn't we all? There's a question here. Thank you.
David, great talk.
Thanks.
So you get many, many ideas, probably all at once. How do you pick what to work on? And then once you start to work on something, how do you decide to cut it off when it's not going in the right direction?
Yeah. Great questions. I see a couple of my students laughing because they know what I tell them in this situation. But what I always tell my trainees, I think there's only one thing that matters about your project. You've got to be really excited about, If you can't wait to come in the morning to see what happened on the gel, then it's a good project. And if you don't feel that way, it's not a good project for you. When to stop, that is always a tough thing. When I give my students and trainees a lot of leeway there. I'll talk to them about it. If I think it's a bad idea or this won't work, I'll try to talk them out of it. But if I can't talk them out of it, I tell them, go ahead and do it anyway. And I have had times where there was something I thought was a complete waste of time, and the trainee was just wanted to do it, and they turned out to be right. So that's humbling enough that we'll discuss it. But it's sort of their call how much time and effort they want to put into it. But it is, it's a huge privilege to be able to mentor these young scientists and watch their careers develop. And I'm really glad to have that privilege.
Great. I'll pass the microphone to you.
Yeah, thanks, Dave. The amount of genes involved in hemostasis is huge, which suggests there's a lot of redundancy. And I'm really fascinated by you saying, too much is not good, not enough is not good, with the assumption that in between those two are normal, but in fact, there could be above normal, which perhaps is what you found with this SURF 4. And so a couple questions. One, do humans have SURF 4 polymorphisms that actually
great question.
Support better outcomes lifelong? And are there other types of similar phenomena in all of the
Yeah,
hemostasis cascade?
No, that's a great question. SIRF4 is more in the cholesterol regulation realm than, but actually in big genetic studies of millions of patients, genome-wide association studies, GWAS, SIRF4 actually does show up. There are some rare variants in there that do have an effect that are correlated with cholesterol. So it looks like variation in humans does contribute. In our experiments, it's mainly complete deficiency, but even the mice that have one copy, there is some change, and that's, I think, what we're picking up in these things. So it does look like that translates. And for the blood clotting proteins, I think it's one of these things that regulation, it's so critical to get that balance right, you know, because we're getting injured all the time and bitten by snakes and running into tigers and all this sort of stuff. And if you bleed too much, it's all over. If you clot too much when that happens, it's all over. And there's also this race going on all the time with infectious organisms, bacteria that are going to come in, and your blood clotting system walls off that bacteria so that it can't spread everywhere. Srishti Bay in my lab is a microbiologist, came to the lab. I love getting people from totally different fields, and she works on how that all works. Streptokinase, a protein from Streptococcus that's critical to its to how it makes you sick. It has to be able to dissolve the blood count. So it has to be perfectly, that's why it's such a complex thing, and in different ways, under different circumstances, you got to fine tune it just right. And evolution, it's been a very strong force throughout evolution to get it to this precisely tuned system. And, and you can tolerate a fair bit of variability, other things compensate, but, it's millions of years have gone into getting that. I don't know if it's just right, but it works for now.
Our last question in the back there. Great, the microphone
Joel.
is coming to you. Thank you.
David, fascinating talk. You mentioned having people come in your lab from different fields, and I think one of the great strengths of your work and one of the great advantages of working in an environment like we have here is we got people who do all kinds of different stuff all over the place in this room, in your lab. My question for you is, what discipline have you not had somebody work with in your lab that you would love to have them come to your office tomorrow morning and say, hi, can I work in your lab?
That's a really good question. I have to think about that some more. Maybe we'll poll the lab about this tomorrow. Who do we? I tell you, I got to say, not too much of an advertisement for the Life Sciences Institute where my lab is, but what I love about it is we have people from all different disciplines. Upstairs for me, Dan Kleonsky's in biology on main campus. Downstairs for me, David Sherman's in the pharmacy, is in pharmacy school. Anna Mapp's in chemistry. We have people from all different, and I love colliding with them. Years ago, we solved the structure of von Willebrand's factor and factor VIII, which formed this very complicated complex using cryo-EM. I'm embarrassed to admit I didn't even know what cryo-EM was, but there was a guy who was right upstairs from me who did cryo-EM and bumping into people at the, I think being in an institution like ours, where we've got everything from a spectacular engineering school to the dental school you run into people who do totally different things, and that's incredibly fun and exciting, and just brainstorming, you come up with ideas. And we've had collaborations and done projects like that with lots of other people in the LSI from all different departments. And it's a great thing. I mean, it is a huge plus of a university like ours, as opposed to being in an isolated medical school or an isolated, that everyone's here, and those kinds of ideas of stuff that you never would have thought of. So the next idea like that, the key is I haven't thought of it yet. And it may be at the water fountain or over a cup of coffee at a faculty meeting that it'll be, oh, gee, we could do this, And that, again, it's the incredible privilege we have of doing this stuff. It really is enormous fun. We're very lucky.
Well, thank you very much, Professor Ginsburg. Please join me in thanking our Henry Russel Lecturer.